Biological immortality is a concept that fascinates many people. While death and aging are inevitable for most living beings, certain species seem to have found ways to live indefinitely without experiencing the effects of aging. Biological immortality is defined as a state in which the rate of mortality from senescence is stable or decreasing, thus decoupling it from chronological age. Biological immortality is a rare phenomenon observed in some organisms, such as certain species of jellyfish and turtles. These organisms can avoid death from senescence by continuously regenerating their cells and tissues. Biological immortality, sometimes referred to as bio-indefinite mortality, refers to the ability of an organism or a cell to avoid death caused by senescence or disease, allowing them to continue living and dividing indefinitely. Biological immortality is a state in which the rate of mortality from senescence is stable or decreasing, thus decoupling it from chronological age. Various unicellular and multicellular species, including some vertebrates, achieve this state either throughout their existence or after living long enough. A biologically immortal living being can still die from means other than senescence, such as through injury, poison, disease, predation, lack of available resources, or changes to environment. It has always been believed that normal human cells are biologically immortal until the discovery of the Hayflick limit. The Hayflick limit is a concept in cell biology that refers to the number of times a normal human cell population can divide before it stops dividing and enters a state of senescence, or cell death. For most human cells, this limit is thought to be around 50 to 70 cell divisions. An important factor limiting the lifespan of cells in vivo is the Hayflick limit. It was first discovered in 1961 by Dr. Leonard Hayflick while studying cellular senescence. This restriction was later found to be due to the shortening of telomeres, the protective caps at the ends of chromosomes, after each cell division. However, the Hayflick limit is neither the end of biological immortality, nor is biological immortality science fiction. Some types of cells can divide infinitely without hitting the Hayflick limit. Biological immortality is an objective existence on Earth. Now let's look at some examples from biology. Germ cells are considered to be immortal cells because they have the ability to divide and differentiate into eggs and sperm throughout the lifespan of an organism, allowing genetic information to be passed from one generation to the next. Germ cells are set aside early in embryonic development, during the gastrula stage, and are the only cells capable of undergoing meiosis as well as mitosis. In contrast to somatic cells, which form the building blocks of the body and have a limited number of cell divisions, germ cells are not subject to the same constraints and can continue to divide and differentiate throughout an individual's lifetime. Stem cells have been described as having biological immortality because they are capable of unlimited self-renewal and can differentiate into multiple cell types. This immortality is due to increased proteostasis that controls the quality of proteins within the cells. Additionally, stem cells have protective caps at chromosome ends called telomeres, which allow them to continue dividing and replicating without undergoing senescence or cell death. However, it is important to note that stem cells do undergo aging, and while they may not age in the same way as other cells, they can become less efficient over time. On the other hand, cancer cells are also capable of dividing infinitely without encountering the Hayflick limit. Cancer cells are often described as biologically immortal because, unlike normal cells, they can multiply and divide indefinitely without aging and dying. They do this through mutations in genes that control cell division and replication, which allow them to continue dividing uncontrollably. Specifically, a genetic mutation triggers the production of an enzyme called telomerase, which prevents the shortening of telomeres, the protective caps at the ends of chromosomes. Telomere shortening is a natural part of the aging process, limiting the number of times normal cells can divide before dying. However, telomerase allows cancer cells to continue dividing unchecked, leading to tumor formation and potential metastasis. Cancer is essentially a disease of uncontrolled cell division, and the occurrence and development of cancer are related to changes in the activity of cell cycle regulators. Certain unicellular organisms, such as bacteria and yeast, can also achieve biological immortality through symmetrical division, because symmetrically dividing bacteria and yeast do not experience senescence like multicellular organisms. Aging in multicellular organisms is often associated with DNA damage, mutation accumulation, and cellular senescence. In contrast, single-celled organisms such as bacteria and yeast divide symmetrically, meaning that both daughter cells receive equal amounts of cellular components, including undamaged DNA, during each division. This means they can theoretically divide indefinitely without accumulating age-related damage or mutations. It is worth noting, however, that bacterial and yeast cells can still die from environmental factors such as nutrient depletion or predation, and accumulate damage over time, leading to reduced reproductive capacity and eventual death. Hydra is another example of an organism that demonstrates biological immortality. These small freshwater creatures can constantly produce new cells, allowing them to rejuvenate and stay young forever. They don't seem to age, suggesting they may be immortal. Hydras are a genus of the Cnidaria phylum. All cnidarians can regenerate, allowing them to recover from injury and to reproduce asexually. Hydras are simple, freshwater animals possessing radial symmetry and contain postmeiotic cells, cells that will never divide again, only in the extremities. All hydra cells continually divide. It has been suggested that hydras do not undergo senescence and, as such, are biologically immortal. In a 4-year study, 3 cohorts of hydra did not show an increase in mortality with age. It is possible that these animals live much longer, considering that they reach maturity in 5 to 10 days. However, this does not explain how hydras are subsequently able to maintain telomere lengths. Turritopsis dohrnii and Turritopsis nitricula are 2 different small, 5 mm jellyfish species. In adulthood, a juvenile state of sexual immaturity can be reverted to using cellular transdifferentiation to replenish cells following sexual reproduction. That is to say, they can be rejuvenated. This cycle can repeat indefinitely, potentially making it biologically immortal. These creatures originated in the Caribbean, but have now spread around the world. The discovery of this unique ability has led to growing interest in understanding the molecular and genetic mechanisms behind jellyfish's ability to rejuvenate. The key molecular mechanisms for its rejuvenation appear to involve DNA replication and repair and stem cell renewal, according to a comparative genomics study. Immortal jellyfish aren't the only species with this ability. Others include Laodicea undulata jellyfish and species in the genus Aurelia Scyphozoan. Planarian flatworms are known for their ability to regenerate, which has led to research investigating their potential for biological immortality. Planarian flatworms reproduce sexually and asexually. Planarians are able to regenerate all body parts, including heads and brains, from small fragments of their bodies. In fact, some studies suggest that planarians may have infinite lifespans, as they show no signs of aging and are able to constantly regenerate cells. Studies of the genus Camellia have shown that these planarians appear to be able to regenerate, i.e. heal, indefinitely, and that asexual individuals possess, apparently unlimited, telomere regeneration capacity, supported by large numbers of highly proliferative adult stem cells. Both asexual and sexual animals show an age-related decline in telomere length. However, asexual animals are able to maintain telomere length on somatic cells, i.e., during fission reproduction or when regeneration is induced by amputation, whereas sexual animals in restoration of telomere reproduction by elongation during sexual behavior or during embryogenesis as in other sexual species. Steady-state telomerase activity observed in both asexual and sexual animals is insufficient to maintain telomere length, while regenerating asexual animals the increased activity in the is sufficient to renew telomere length. While technically not biologically immortal, planarians are able to continually renew cells and regenerate lost body parts, making it possible for them to live indefinitely without signs of aging or reduced ability to regenerate. This has sparked interest in studying planarians as model organisms for regenerative medicine and potentially unlocking the secrets of biological immortality. For sexually reproducing planarians, individual planarians can live up to 3 years, probably due to the ability of new cells to continually replace older cells. And for asexually reproducing planarians, individual animals in clonal lines of certain planarian species that replicate by fission have been maintained for more than 15 years. In humans, achieving biological immortality is currently not possible, but research is being conducted to understand the aging process and develop interventions that can delay or prevent age-related diseases.